Imagine running a microbiology test and every result you give is 100% reliable. But how? By using the magic guardians of every lab, that is the quality control strains. Hello everyone, welcome back. And today we are going to talk about something that makes every microbiology result trustworthy. That is the QC strains used in routine laboratories. First of all, let's discuss what are QC strains. The QC strains are standard reference microorganisms with known characteristics. They help us verify whether the media is working properly, the biochemical tests are performing correctly, the antibiotic susceptibility discs are accurate, the strains and reagents are functioning, and the instruments are reliable. These strains act like internal controls for the lab. The question is, why are QC strains important? Because they ensure accuracy, precision, reducibility, and the validity of reports. In simple words, QC strains protect the reputation of the lab. Before moving to the important QC strains, let's discuss about ATCC. ATCC stands for American Type Culture Collection. Here you can see the examples of ATCC numbers that are commonly used. These are the world's largest and most trusted organization that provides standard reference microorganisms used for research, diagnostics, and quality control. Let's discuss about the most important QC strains that are used in microbiology. The first category is gram-positive cocci. The organisms include Staphylococcus aureus, Staphylococcus epidermis, Enterococcus faecalis, Streptococcus pyogenes, and Streptococcus pneumoniae. These Staphylococcus aureus are used for antibiotic sensitivity QC and biochemical test. The Staphylococcus epidermis plays vital role in coagulase test and mannitol test QC. The Enterococcus faecalis, which plays main role in antibiotic sensitivity QC and bile esculin QC. The Streptococcus pyogenes for bacitracin sensitivity QC and the Streptococcus pneumoniae for octocin sensitivity and bile solubility QC. The next category is gram-negative bacilli. The organisms include E. coli, Klebsiella pneumonia, Pseudomonas aeruginosa, Salmonella typhimurium, and Proteus mirabilis. The E. coli is used for antibiotic sensitivity QC, indole and citrate QC, Klebsiella pneumonia for biochemical QC, and Pseudomonas aeruginosa for antibiotic sensitivity QC and oxidase QC. The biochemical QC and the media QC, especially XLD, can be performed using Salmonella typhimurium. The Proteus mirabilis plays vital role in the swarming test and urease QC. The third category is fungi. The organisms include Candida albicans, Candida tropicalis, and Aspergillus niger. The Candida albicans used for germ tube QC and chromaga QC. The sugar assimilation test can be performed using Candida tropicalis. And the Aspergillus niger plays vital role in antifungal media QC. The fourth category is Mycobacteria. The organism include Mycobacterium tuberculosis and it's used for culture QC and drug susceptibility QC. Let's see how the QC strains should be used. Grow the QC strains on the same media you use for patient samples. Compare their reactions, especially the colony morphology, pigmentation, biochemical result, zone diameter, and their growth patterns. If QC behaves exactly as expected, the media or the test is valid. If not, then reject the batch or recheck or troubleshoot. The precautions while handling the QC strains are vital. Use the authorized ATCC strains only. Store in proper conditions, especially in freeze-dried vials. Maintain strict aseptic technique. Record in QC logbook daily. Never mix up patient strain with the QC strains. A lab without QC strain is like a report without trust. So, always let your QC strains speak for the accuracy of your work. Thank you for watching. I'm Sneha signing off for today. See you in the next powerful microbiology video only on Medical Guruji. Until then, stay curious, stay accurate. Thank you and bye bye.